Good afternoon, everybody. Thank you for coming to this session that was prepared especially for you from the Graduate Student Committee. So first of all, I would like to give a warm welcome to everybody sitting here and also to our global audience watching the live streaming of this session. So 2 reminders for the audience present in the Convention Center. Please turn off your cell phones. So this is an opportunity for you to look at them and make sure that they won't ring in the middle of a talk. And also, we ask that you refrain from taking pictures, so no photography allowed. Now, from the speakers, I'm going to ask 3 things. Remind you of 3 things. The first is that I will stand up after 25 minutes, and that should give you about 1 minute. Maybe I'll stand up at 23 minutes, and I'll give you about 2 to 3 minutes to finish your talk. The other thing is we can't use a pointer because we are streaming this, and so if you need to point at something, use the mouse. And the third one is when people ask you a question here in the audience, please repeat the question before you answer it. So our first speaker today is Michael Kwabena-Osei. He is coming to us from the CSIR Crops Research Institute in Kumasi, Ghana. And the title of his talk is Capacity Building of Tomato Farmers in Ghana, the Case of IPM Package.
Thank you. As she rightly said, I am Michael Kwabena-Osei, coming from the CSR Crops Research Institute, Kumasi. In the next few minutes, I'm going to present to you my work on capacity building of tomato farmers in Ghana, the case of IPM package. Working in collaboration with Sally Miller from Ohio State University and Robert Gibson from UC Davis. Receiving funding from USA through Virginia Tech. Excuse me. Okay. Sorry. As an outline of my presentation, we're going to briefly look at some introduction to the work and, of course, the capacity building that took place using the IPM package and some results arising from building the capacities of farmers. As a way of introduction, let me say that tomatoes are important for their nutritive values, income generation, and of course, economic value and impact on the environment. In Ghana, tomato is consumed in almost every household daily. They are used in the form of stews, soup, salad, and what have you. Now, despite the importance of tomato, the crop is posed with a lot of constraints, and one of which is pest and disease. Among the pest and disease, tomato yellow leaf curve viral disease, which is of economic importance, bacterial wilt, nematode, among the rest. Then secondly, misuse of pesticides, lack of up-to-date technology, and lack of high-yielding varieties among the production constraints. Previous work has made some progress in addressing some of these constraints. It is, however, not possible, to depend solely on a particular method of controlling this disease. Efforts must therefore be taken to keep plants from becoming infected, of course, to manage the rate and the timing and severity of infection to protect this crop. The other concerns like low awareness of these pesticides, and which has increased their misuse and abuse. And the misuse of these pesticides have also raised some concerns about health hazards, death, and environmental pollution. There is therefore the need to put in place some important strategy to minimize this pest pressure, frequency, and the rate of pesticide application to protect man and the environment. As you can see this slide, this is typical of some of the farmers in northern Ghana who spray their tomato fields without covering themselves. This man is not wearing any Wellington boot, neither is he wearing any top or something to cover the nose. So you ask him, are you safe? And he tells you, I don't know. It tells you he's ready and willing to learn. So that takes us to our capacity building. So we look at what going into the capacity building using IPM package. And before we start, our collaborators join us in Ghana to look at some of the farmers' programs in terms of the tomato disease on the field, talking about bacterial warts, talking about TYCV, CMV, TMV, among the rest. We had a chance to also interact with the farmers to know some of the problems they are going through in terms of the use of the pesticides, as you can see on the slide here. So here you see Bob Giverson showing how to use immunostrips to detect CMV and TMV specific. And in that case, once we get to know the incidence is high, then we can chip it into our IPM program. So here in Ghana, we have 10 regions, and out of the 10 regions, we build capacity of farmers in 3 of these regions. The Upper East, where we selected 3 locations, the Brahafo region, where we also selected 2 locations, and the Ashanti region, where we selected 2 locations. And these 3 selected regions are translated into the agroecological zones in Ghana. These are based in the climate and soil types. So what goes into our IPM package? Of course, we look into site selection as to what farmers should select their site for tomato, what they should look at it. We look at site preparation, what should go into the preparation. And I might say the idea behind the IPM is to reduce use of pesticides. So at the end, farmers can save money, reduce cost of production, and, be attractive to get profit. So we look at what goes into the seedbed preparation or the nursery, the transplanting, the production, and post-harvest. So with a combined IPM package, at the site selection, we advise farmers to go for land that has not been used for tomato production in the previous years. And the rationale is that the pest buildup in this land will be reduced. Again, we advise them to site their tomato production, not to site their tomato production next to an old farms. Because once you do that, as soon as you establish your new seedlings, the white flies or other pests are likely to move to your new seedlings. So we advise them as much as possible not to site their new tomato seedlings near to an old farm, and if possible, not to also site near to other solanaceae crops like pepper and garden eggs that share common pests with tomato. Now, if you look at before growing season, as part of our combined IPM package, I look at white fly transmitted viruses. As mentioned, we advise them to use resistant cultivars. And as you go further, you get to know some of the resistant materials as far as virus or TYCV is concerned, which we promoted to farmers, and to avoid planting new fields near other fields, as already mentioned. And during the growing season, there's one thing we learn about farmers. They use what we call calendar spraying. They establish their field and they start spraying whether they've seen disease or not. But IPM introduces what we call monitoring. You have to reach a threshold before an action is taken. And once you do that, you realize you don't use too many pesticides, and your cost of production goes down, and you end up getting good and healthy plants with more profit. Then we explain to our farmers, if you see symptoms of tomato yellow leaf curve virus, at the initial stage, you try to root them off, because the assumption is that the white flies you see them on the field, not all of them are carrying the inoculum of the virus. So once you see symptoms of one of the virus plants, you rogue it off before the other white fly that are not having a inoculum feed on it and transfer it to the other plants. So these are some of the teachings we to the farmers through. We also realize that after growing the plant or after harvesting, most of the farmers leave their fields bare. They finish harvesting and they don't care what happens. But we explain to them, once you do that, you'll build up pests. So the best way is to destroy the field, remove all the crops after harvesting, so that next planting, you don't see those pests again. Now let's look into what goes into the tomato IPM technology. The chart you see here, we are concentrating on the avoidance using of tolerant or resistant varieties, and as you go further, you see those varieties we use. And here, we are promoting action threshold. That is, farmers need to monitor, and we to them through before they apply any chemicals. And even with the insecticide that they have been using, we advise them to be alternating them. Because if we tend to use one particular insecticide for a long time, then the white flies or the other pest tend to be resistant to those insecticides. So once you alternate, then when they are expecting you are bringing this particular insecticide, they get confused because you change it. So these are some of the things we take them through. And of course, sampling of the white flies. Now we go into the IPM package deeply. With the first selection, as already indicated, as much as possible, we look for fields that had not had tomato in the previous years and not too close to another farm, as already mentioned. And importantly, observing a host-free period. And this, I must say, really worked in northern Ghana, where in the rainy season, they can't grow tomatoes. So for 2 or 3 months, there will be no tomatoes. That helped them to observe a host-free period. So that when it's time for the actual tomato growing season, there's less incidence of the whiteflies and less incidence of the virus. Sorry. Then with the cultivar selection, we had a privilege receiving 4 varieties from the U.S., 2 hybrids, Shasta Enhanced, and 2 open-pollinated, OPB 144, OPB 155, and we recruited a local into our tribe. And what happened was that we took farmers through the seedbed preparation. Amazing. For each location, we allow farmers to do their own way of nursery, and we also indicated to them what the IPM nursery should look like. And for the farmers, the tomato farmers in Ghana, most of them will want to make their bed 1 meter by 10 meters. Just raise the bed and put some chemicals in the nursery. They also understand there are some pathogens in the soil that can disturb them and just grow. And these chemicals are not free. They are very harmful because, as you saw in the picture, they don't protect themselves. So we thought we should introduce what we call sterilization. And as we go further, you see how the IPM bed sterilization looks like and the results we had. So at each location, we plowed the land harrow, and we had 2 plows. We had a farmer's practice and the research, or the IPM practice. And we have 5 varieties, as already mentioned, and we replicated them in RCBD, 5 replications. So in the seedbed preparation, as already mentioned, farmers will make their normal bed, put some chemicals, and nurse their seeds. But we introduced this sterilization, where we made the bed, sprinkle some water on the bed, cover with some dry straw, as you see here, then we set fire. So the idea is that you want to kill pathogens, and this time you don't use chemicals. Sorry, I want to go back. Okay. So the idea is that you want to kill the harmful pathogens in the soil so that you reduce the use of chemicals where our farmers cannot protect themselves and where it costs them money to do. And this is very simple. So once you sterilize and you allow the fire to be on there, after some time, that is, if you do in the morning, you can go around in the evening. That's when the sun is down. Then you can start nest your seeds. And as you can see here, the beds were sterilized to help eliminate the soil-borne diseases and pathogens, and seeds were then planted 1 centimeter apart and 2 to 4 millimeters deep. Again, when it came to nesting the seeds, most of the farmers in Ghana still continue to do what we call broadcasting. And you agree with me, if you are using hybrids, which are quite expensive, and you are broadcasting them, it means you are going to waste a lot of the seeds. It's going to cost you money. And again, when they emerge, you see that they don't look healthy because they compete on that piece of land. So we took our time to take them through what we call drilling, as you can see here. This time we made drills and explained to the farmers not to even broadcast, but again, we allow them to broadcast, and this is the IPM we drill and we dictated to them. And again, when it comes to the number of seeds you should put in, for those farmers who could agree with us to drill, if you allow them to put seeds in one particular drill, they still put in a lot. But we have done it at the research station, that if you just take a cork of beer or Fanta and you fill it, you should be able to spread it evening. Sorry. You should be able to spread it evening, and they come out nicely than just making the drill and putting any quantity of seeds. So those were some of the training. And at each stage, we also explain to our farmers the rationale behind whatever we are doing, so that at the end, they will not just take it, but they will take it to their understanding and with all the joy. And at the end of the nursery, we cover with palm fronds and water again. Then after 5 days, we can go and check if they are in. But usually after the 5 days, when the seedlings emerge, where we have high incidence of the whiteflies, we do cover with nets. The idea is to prevent the whiteflies from transmitting virus. Studies show that early infestation of virus to our seedlings are likely to cause about 75 to 100% losses. And what we know is that if they are infected at the nursery, when you transplant to the field, between 2 to 3 weeks, you begin to see symptoms of the virus. And that time is too late for you to spray insecticides. And that is what our farmers didn't know. they mix chemicals, and it is when they see symptoms that they think they can do that. But we explain to them that if you're able to escape the whitefly from infecting them at the nursery stage, then you can do a lot when you transplant. You can overcome them. So you see that these are the same varieties, but this one is coming from the farmers' nursery where they broadcasted. They were nursed the same day. This is coming from the IPM. So when the seedlings were ready, we asked them to pull. They saw, they believe. You see, so this is not something we're forcing on to them. So once they've seen and believe, then gradually they begin to practice it. So that's the field layout for transplanting. So on the field, we have the farmer's field where we transplanted seedlings from the farmer's nursery bed where they broadcasted, and the research of the IPM where we transplanted seedlings from the IPM nursery bed. Now, when we transplanted, we had to be on the field every 2 weeks to take farmers through what has been worrying them, the kind of things they see that they begin to, spray different insecticides, mix them and the rest. And we explain to them at what stage you have to spray, threshold that you have to reach before you spray. No need to use calendar spray and be spraying when you have not seen any symptoms of the disease, so that, as already indicated, it will reduce the cost of production and get high profit. So what goes into the IPM or the research field? We monitor for pests and disease, and of course, we encourage using spraying, recommended clothing for spraying. And at the early stage, when you see symptoms of virus, especially TYCV, you rogue them off to prevent the other whitefly from feeding on it and transferring it into the others. The farmers field, they were using calendar spraying. We allowed them to do that so that we could compare the results of what comes from the IPM and the farmers. So harvest period, we harvest the research field differently from the farmers and compare the yields. And again, we advise that they destroy their old crops or burnt after harvesting. And I've explained that. So you can see a session of our farmers where our collaborators join us and we have pictures together. And these are some of the fruits here. So briefly, let's look at some results. So here we look at some performance of the 5 tomato varieties under the IPM and the farmers field at one of the locations called Via in the Upper East region. So we have 5 varieties, as already mentioned, Shasta and Hens from the U.S., hybrids, and these are open pollinated, and we included a local. So here we are interested in looking at, in fact, we brought in the aspect of aphids because we realized that at all the locations where we cited our trials, we had high population of aphids. So we took the advantage to compare number of plants infested with aphids on the IPM fields as against the farmers' practice. And you realize that we recorded percentage decrease over the farmers for as far as the number of plants infested with aphids are concerned. And if you take Shasta, we recorded 54.64% decrease over the farmers' practice. If you go to the number of fruits bora per plot, the same thing also happened. We had a farmer's practice recording high number of fruits bora per plot compared to the IPM, giving us percentage decrease over the farmer's practice in all the varieties. Similarly, in the fruit weight, or let me talk about the number of fruits per plot, because our farmers in Ghana prefer to use closer spacing, thinking that when they use closer spacing, they will get more yield. They forget about the fact that the more they are closer, the less your fruit weight also happens. So if you look at the number of fruits per plot, of course, when they have closer spacing, they are recording higher number of fruits per plot than the IPM. But this was not translated when we got to the fruit weight, where the IPM was recording higher than the farmer's practice, because the IPM encourages bigger spacing, 100 by 50, where farmers want to do closer spacing. So again, we allow them, they see, but the funny thing is that in Ghana, we don't sell by weighing, unlike the other European country and probably US here. We sell by the size and grouping them. So again, the issue of preaching about spacing here is important, because if I go to the market, I'll prefer to pick the bigger ones. So once you adopt the IPM spacing, wider spacing, then you tend to have bigger fruits than having many fruits, and they are very small. So that is what we preach here. So if you look at the IPM, the fruit weight, we recorded percentage increase over all the farmers' practice. And it tells you using a bigger spacing or larger spacing. Again, another location, turn on, the story is not different. In all the varieties, in terms of the number of plants infested with aphids, we also recorded percentage decrease over the farmers' practice. Similarly, in the number of fruit borers per plot, the farmers' practice had higher number of fruits per plot, giving a percentage decrease over the farmer's practice. Again, as already explained, the number of fruit per plot, farmers are higher number of fruits per plot because of the closer spacing. But again, this was not translated in fruit weight, where the IPM had higher fruit weights over the farmer's practice, recording percentage increase over farmer's practice. In conclusion, let me say that by implementing this IPM package in Ghana, farmers' capacity, of course, not all the 10 regions, but the 3 regions cultivation capacity in tomato cultivation are built. What we need to do is to rather scale up this technology to the other regions which we're not able to cover. And there is also a high probability that effective management of any tomato infecting virus, like tomato yellow decay virus, can be accomplished with this IPM technology. I can't sit down without acknowledging the following. Of course, the IPM Innovation Lab making it possible for me to come here and funding this work, and our tomato farmers who avail themselves to be built. And of course, the project team, I didn't do it alone. I had other local project team members who supported. And more importantly, our collaborators from the US. Thank you very much for your attention.
Any questions?
Are the farmers in Ghana mostly men or mostly women?
That's a good question. We have a mix, but you have a lot of the women doing a lot of the work. Yeah.
The ones who do the work are the women, right?
It depends on the activity. For example, when it gets to harvesting, you have a lot of women helping the harvesting and marketing of the crop. Yeah. All right.
When you start a project, is it easy to get farmer collaboration, or do you have to go out and really encourage them to be a part of the group?
Yeah, that's a good question. In Ghana, I'm coming from the Ministry of Environment, Science and Technology. We had a Ministry of Agriculture where we deal with the extension officers. So when we were starting, those locations that we selected, we visited the Ministry of Agriculture to give the extension officers who are working with the farmers. And they introduced the tomato farmers to us, and we started building the rapport from there. Yeah.
What is generally the farm size? How big
Yeah. It's, you want to know the general farm size of an average farmer. It's less than acre, 1 acre. Yeah.
Is it pretty common for farmers in the region to have access to things like improved seeds and water and chemical inputs?
Yeah, when you talk about water, you know, tomato does, you want to know whether farmers have problem getting access to seeds, water, etc. In fact, you agree with me that tomato, especially where I'm coming from, does very well in the dry season. But our farmers are constrained when it comes to irrigation. And because of that, Burkina Faso, another neighboring country, takes advantage during the dry season to, you know, bring in tomatoes to our country. So farmers are limited when it comes to irrigation. And because of this problem, they all want to crop in the rainy season. And not all the varieties do well in the rainy season. So we tend to have a lot of blights. When it comes to varieties, the crop research institute where I'm coming from, and even with this project, we're doing everything possible. The farmers selected some of these varieties, which we're doing possible to make them available. But again, they depend on some of these introduced hybrids from other countries. But because good trials are not made, they come in the beginning of the year, they may be doing very well, but in the second year, they succumb to some of the diseases in Ghana. Yeah, thank you. Yeah.
I noticed that the yield of the open pollinated varieties on the whole are higher than the hybrids. Is it taste or why does anybody bother to plant hybrids?
You see, the issue with hybrid, for example, in our case, we're getting the hybrid from US for the purpose of the project, you know, to try. So if farmers select the hybrid, then they're going to find a problem getting back because they can't recycle the seed. They need to come back to the source. So they prefer one of the OPs 155 where they can recycle. But again, we have to take our time to take them through how to select healthy seeds. Otherwise, you recycle disease and the rest. Yeah.
How long does it take for, in average, for the farmers to accept your recommendations?
Yeah, you want to know how long it takes for the farmers to accept our recommendation. In the extension, they have what we call laggers. Excuse me to use this. You have the early adopters. You see, these were different locations that were selected. For some locations, they are early adopters. Early adopters here, I want to make tomato takes about 3 months. So you finish the season, if they are convinced, after the 3 months, they accept it. Where you have laggers, they want to see other people making progress with the technology before they accept it. And for those people, it can take a year. Yeah, thank you.
You didn't mention the amount of virus that were in those plots. Does that mean that the virus pressure wasn't high in your plots?
So which virus are you referring to?
TYLCB. You didn't show any data for TYLCB incidents in the IPM plot versus the farmer field. So what was the situation there for TYLCB?
Yeah, Bob wanted to know why I didn't show data for TYLCB. If you take a place like Northern Ghana, the upper is where I presented. Because this package really worked well, as we speak, the problem with TYLCB has gone down. Because they practice a lot of host-free period, 2 to 3 months, they may not be growing the variety in that season. Therefore, when they grow, they're able to escape some of this virus. But if you take a place like the Ashanti region of Bunahafo, because they want to grow tomato in the rainy season and in the dry season, they tend to have high incidence of the disease. Now, coming back to the question, this was a team where different groups were taking different data. And I thought I wasn't having the whole time here to present everything. But we do have data for that. And Bob is part of the team and he's aware. And, I switched to number of infested aphids because I did mention, whilst we were taking data for the whiteflies, for the virus, we saw high incidence of the aphids. And in fact, at one point in time, we had to check with immunostrains to find out whether these aphids were causing the CMV, alongside with the TMV. So truly we have data for that, but I thought I should restrict myself to this because of time. I don't think I can present everything. Thank you.
How well established is the value chain in Ghana for tomatoes during, for example, the low season? Is it easy to take them from farmer to market, and does the farmer get most of the benefit?
Yeah, so you want to know how we establish the value chain as far as tomato is concerned. Yeah, if, our farmers are programmed with marketing, and if I introduce IPM, where we made success in northern region, the question they keep on asking is, now we are practicing IPM, we are producing more. Where do we sell them? So you see. So there are two groups. We have the market queen, those who go to the farmers, buy them in bulk, and take it to the other retailers. Then we have, it's supposed to work, but it's not working, tomato industry, where we expect the farmers to take their produce to those industry. But here, there is no honesty here, because where farmers are not getting market, they take it to the industry. When the industry are expecting them and the farmers get good price, they dodge them. So you see, and signing MOU is not working because the farmers are not well organized. They are not in a situation. So there's actually problem with the value chain. But the current government is making headways in establishing a lot of the tomato industry, reviving those that have been there for now, so that there can be that kind of MOU where farmers can produce and we can sell their produce to the industry. Yeah.
More questions? Okay, thank you.
Thank you.
So in this 1 minute that I have for it to be 1:30, I just want to remind you that we have a 15-minute discussion period at the end of the session. If anybody has more questions for the presenters, that would be the time to make them. And we can also talk about some other things if you want to. Okay. So our next presenter is our first student presenter of this afternoon, and it's Alejandra Huerta from the University of Wisconsin-Madison, who will be talking to us about a window of opportunity summer tomato production in Bangladesh.
Welcome, and thank you all for being here. And for those of you watching the live stream, I'm really happy that you're participating in this. And tweet or Facebook someone in here if you have a question, if you can. So I'm going to start with my title, A Window of Opportunity, Summer Tomato Production in Bangladesh, not only because it reflects the project itself, but because it's also a window of opportunity for me to learn a little bit about what it is to work in a developing country, and what some of these phrases that sometimes go on the introduction of research articles and molecular interaction studies, how does that really apply, and how is that being translated, or if it is being translated. Before I do, though, I have to say that it takes a village. It takes a village to write one of these Borlaug Fellowship grants. It takes a village to actually carry out the work. So I will be saying I, but all of the time I will be referring to we. And that's the Wong Lab right here from the AVRDC, the Asian Vegetable and Research Development Center, and that's the group that I was working with while performing this project. And also my home institution and Dr. Kelly Allen, who supported me in taking the 6-month leave from my thesis dissertation to do it. The University of Wisconsin, of course, for giving me the support. The National Science Foundation for giving me the opportunity to further my educational career. The Bangladesh Agricultural Research Institute, which was a collaborative effort. And ultimately the USAID, who was the main funding agency for this. So the presentation outline, I will start with a why, who, what, where, and ultimately how I ended up at the Borlaug Fellowship. A short introduction into Bangladesh, and then I will get into the gist of the summer tomato production. Food security. When I started thinking about food security was while I was presenting and getting ready for my qualifying exam. And I was reading a lot of introductions, and a lot of the introductions said something like pathogen X threatens crop production X worldwide. And I was just like, all right, we're going to make a difference. And then threatening livelihoods in developing nations. And I was like, how are we directly impacting these developing nations? And one of my favorite is global food production must increase by 50% to meet world's population by 2050. And all of these are statements that are relevant and are important. And I'm not saying that none of them matter. They absolutely matter. But what and how do they matter to one of these nations that we're trying to impact? So I often found that it became a trend, and it was something that, people were going back to reach into a pot of gold for funding. And we all struggle for funding. I know that. I've been there. Doing the Borlaug Fellowship, it was a call. But then I started doing, in that time, some more research, and I ran into this meeting review from MPMI 2009, where a retired professor from the University of Wisconsin was reminding scientists to, yes, do excellent research, but always remember and keep in mind, how is that research going to translate? How is that going to impact it? So that got me thinking, okay, I want to go out there and I want to make a difference, but how can I make a difference if I don't know what the problem is? So what is food security? One of the other really interesting things is that this fact, 20% of the people in developed nations consumes 86% of the world's goods. That 20% is in the United States and Europe. We more or less dictate what, we can take or we can't. And we, to a certain extent, projects like this are trying to empower other countries. And, how we do that is greatly dependent on the goal that we have as people and as a country or as researchers. And then ultimately the definition of food security, which is food security exists when all the people at all times have physical and economic access to sufficient, safe, and nutritious food to meet their dietary need and food preferences for a healthy and active life. So it's not only are we producing enough food, but what are we doing with that food? Is it actually reaching the people that need it the most? Do people have the economic means to purchase it? Do people have ways to get to the market and buy it? Are they eating the necessary nutritious elements? You know, that's something that really, we have to invest a lot of time and knowledge and be there to better understand it. So then, how are we helping in that development, and how can I help most of all? So that's when I stumbled through a friend of mine on the Borlaug Fellowship in Global Food Security. And this is sponsored by Purdue University. They have two very important programs. One is the actual Borlaug Fellowship, where you go to another country to do some research. And another one is a one-week intense workshop where you learn about these topics. But the main goal of the Borlaug Fellowship in Global Food Security is to train the next generation of scientists. They're going to go and are going to impact development agriculture to support the Feed the Future initiative themes, specifically those 17 countries that they're targeting right now. And to foster that cultural, cross-cultural understanding through on-ground collaborations, which are really important, because they don't, when you don't have a really open line of communication between everyone that's participating, you don't know what they want, they don't know what you want. You come ultimately with different goals. And in order to be successful, you must both come together under an agreement, like, you know, you will do this, I will do this, and hopefully this will work, you know. But you have to have that really clear line of communication, and that's one of the main things that I learned during this internship. And ultimately, to work with an international set of partners, we all have different ideas. We are all individuals, but how can we bring that individuality to have a successful outcome? And I think this picture very much represents that. We had a workshop in Bangladesh where people from Africa, Europe, the United States, South America, and Southeast Asia came to try to improve vegetable production in Bangladesh, and not only in terms of breeding, but how to really enforce the value chain, which has a very important impact on the success of a given project. So when I did my Borlaug Fellowship, I contacted Jia Fen Wang at the AVRDC, and this is her right here. She has worked on Ralstonia solanacearum, which is the host, the pathogen that I work with at Wisconsin. And this is Dr. Peter Hansen, who is a plant breeder at the AVRDC. And years ago, they did this worldwide evaluation of an international set of resistant sources to bacterial wilt in tomato. And I really enjoyed that publication because they took germplasm from all over the world and then they sent it out all over the world to see what the yield and how they performed against different pathogens, in particular bacterial wilt, which varies greatly by location. So I contacted her. It was really random. I said, I want to work with Jia Fen. I looked for her paper. I emailed her, and I said, I would like to apply for a Borlaug Fellowship. Would you support me? And it took 3 days, and she replied to me. I also applied to somebody else, and they never got back to me. So you have to try and then see what happens. So now to a short introduction into the People's Republic of Bangladesh. Bangladesh is right here. It's east of India, and it's west of Burma, and it's in the south. You have the Borlaug, the Bay of Bengal. The official language is Bangla. It's gained independence, quite bloody independence from Pakistan in 1971. Its population is 150 million people. It's the area of the country is 148 square kilometers. Poverty, it has a very high poverty rate. About 65 million people, and 45 million people are in acute poverty. 40% of the population is undernourished and 20% is severely malnourished. The malnourished population are usually very young kids and the elderly. The GDP per capita is $2,100. The religion is Islam in its majority, and 48% of its population is actually involved in agriculture. So agriculture plays a really important role in the society. just to give you an idea of how densely populated that is, it's taking half of the United States population and putting it into the state of Iowa. So like I said, vegetable production has been increasing in the last 15 years or so in Bangladesh, and that is this green line that you see. And from that increase, tomato production is actually very, very low, but in the last 5 years or so, it has definitely been increasing. The top 10 commodities you can see on the right, their diet is mostly rice and potato. They have very, very little vegetables, but their vegetables are important, especially tomato, during the Muslim holidays, which are in the summer. And like in the summer months, and in the summer months, it's really hard to have access to tomatoes. So tomatoes are a high-value cash crop in Bangladesh. They're nutritious. Just a few grams can really get you what you need to support yourself. But there's a certain limitation to tomato production in Bangladesh, and it's that they have an ideal growth conditions. They prefer days of 18 to 25 degrees Celsius and nights of 10 to 20 degrees Celsius. So they're okay with the heat, but they really can't really survive in really high temperatures. And they prefer well-drained soils. And during the summer, in Bangladesh, we really don't have a lot of well-drained soils. So it's a window of opportunity, because if you're able to grow tomatoes under these conditions, you can really get a good profit. So winter tomatoes usually sell for about 10 to 15 cents per US dollars per kilogram, but in the summer months, tomato can be sold for 50 to 80 cents a kilogram, and that's a big difference. And in the summer months, remember, there's not a lot of vegetable production. You have limited access to some vegetables, so this increase in income can really impact a family. Coming back to the constraints of growing tomatoes in the summer, so here we have precipitation on top and temperatures on the bottom. And if you look at the summer months, starting maybe April to September, you can see that precipitation really increases. And also heat. Heat goes from about, say, 24 to 28. It can go up to 35 degrees Celsius, which are not the ideal growing conditions for tomato. And then you have really high precipitation as well. With high precipitation, you don't get those well-drained soils like you see in this picture. And with high temperatures, what happens is that you don't get flower set. The flower aborts, and therefore you don't get any fruit. There are biotic constraints as well during the summer. You have fungal, bacterial, viral, and insect pests. Some of the more important are early blight and black leaf mold, bacterial wilt and bacterial spot, tomato yellow leaf curl virus, and the tospoviruses. And the two major insect pests are tomato leaf miner and tomato fruit borer. So there's also a constant need of fungicide and chemical application in general. But the Bangladeshi are very resilient, and they have found a very successful way of growing summer tomatoes, as you can see in this picture. They use bamboo and plastic to do a very rudimentary shelter. It's not a greenhouse. It's nothing close to it. It's just a shelter. Here you see this young man applying a hormone to the flowers, and this hormone impedes the flower abortion. However, you need to apply this hormone every 2 to 3 times a week in order to get a good yield at the end of the season. So we know that in Bangladesh, some producers are good at, they have the technique down. They're able to grow summer tomatoes, but we really don't know how well rain shelter and grafting or any other technology for that fact leads to the impact in terms of how much money are they getting back at the end of the season. They definitely do a lot of input. How much are they getting back? It's not really known. So we wanted to do a couple of a test with this study, and that was to measure the impact of rain shelter and how much does that impact yield. And can we get a good yield without using rain shelter? And then we have different varieties developed in Bangladesh. And are some of these varieties better for growing during the summer months? And one of the new questions was, is grafting essential to actually get a good yield in the summers? To give you an idea of rain shelter and the cost of it, $1 equals 78 taka. Keep in mind the GDP of the country. In 2012, here we list the unit price of the material needed to build one shelter, and one shelter is 1 decimal, which is pretty much 46 square meters. So you can see that at the end, you have a total $63 to build one shelter during the 2012 growing season. But when we look at the 2013 growing season, the total to build one of these rain shelters is $82. That's a $33 increase, and that's very substantial for a Bangladeshi farmer. And usually you're not also planting just 1 decimal. You have 4 or 5 decimals, so it's quite an investment. And we can also take into account the cost of grafting. To graft one tomato, it takes about 0.05 cents, which is very minimal for us, but for them is 4 taka. And to get one seedling without grafting is 1 taka. So that's a big difference as well. So grafting is important in Bangladesh, and in particular in this trial, because we take rootstock, which is from eggplant, and eggplant is much more resistant to flooding soils, so they perform much better under flooding soils. We take this rootstock that is also highly resistant to some of the soil-borne pathogens that are a problem in Bangladesh. We take the tomato variety of our choice, and in this case we had Bari Tomato Hybrid 4 and Bari Tomato Hybrid 8 that are 2 favorites in the Bangladeshi community. We graft them, and we were grafting them via rubber tubing, a new technique for Bangladesh as well, and then we left them out for in a healing chamber. Grafting is not new to Bangladesh. It's not a new technology that I was taking there. They've been doing this for a really long time, but they are used to doing it with clips and using much larger plants. So it takes a longer. It's more labor intense to do the graft tubing. They also use native solanum species that is thorny to graft onto and use as a rootstock. However, this rootstock in recent years has developed susceptibility to bacterial wilt, which is one of the major problems. So this is a picture of what the rubber tubing in a successful graft looks like in the field, and in time this rubber just decays and falls into the soil, so you don't have to go back and actually remove the clip. So then there was 3 aims to this study. One was to measure the effects of grafting and rain shelter on tomato yield. The second one was to measure the cost and benefit of growing summer tomatoes using grafting, rain shelter, and both. And ultimately to measure disease pest pressures on tomato plant grown under rain shelter and grafting. We had 3 locations. One was in Shanghua, Taiwan, headquarters for the AVRDC. One was in Gazipur, Bangladesh, and the third was in Jeshore, Bangladesh. This is the central of the country. We have really clay, heavy soils there. There's very little tomato production. But here in the southern area, this Jeshore, it's considered the vegetable belt of Bangladesh, and a lot of vegetable production is there, and the farmers are incredibly well adept at growing vegetables. We had 4 reps, 24 plants per treatment, and 8 treatments overall. And then the data collection. I was pretty much the hubble for the data collection, and that's where the communication really came to be of great importance to me and to my partners. Dr. Su-Hong Ling from the AVRDC was the main person monitoring the trials at the AVRDC. Sasib Biswas was a graduate student at the Bangladesh Agricultural University, and he was working on this for his master's thesis, and he was responsible for taking care of the Jeshore and Bangladesh trials. And then I bring this because we had these temperature humidity sensors to actually take data for us. And then we had these sensors that I call with appropriate technology, because we had the sensors, but we didn't have enough money on the budget to buy the sensors to cover it, to keep it from getting wet. But we needed to keep it from getting wet because it's the rainy season. So we sort of brainstormed with my colleague, Sarjeep, and he was just like, oh, let's use these, these plastic bottles and cut the bottom, and then we have a little shelter. And I was like, there we go, we're thinking. So it worked. At the end, it worked. We have to, use what was available to us. So now to get to the data. So this is from our trial in Gazipur, Bangladesh. On the y-axis, you will see yield per metric tons per hectare. And here you have grafting, yes, yes, and rain shelter, yes, yes, and in between, no. So first, our treatment A was with grafting and without grafting, and yield was about 67 metric tons per hectare. When we only had grafting and no rain shelter, there was a substantial decrease in yield. When we had rain shelter and grafting, there was a slight increase than when we only had grafting, but it was not as high as when we had both treatments. And once again, when we had grafting and rain shelter, yield was really decreased. So ultimately, from the Gazipur trials, we were able to see that indeed grafting and rain shelter really did have an impact on tomato yield. And once again, I would like to bring your attention to what the field trial looked like in Bangladesh at this site. And you can see that some of the plastic is coming off from the shelter. And this is not atypical. This is something that's very common. So then you have to get additional farmers to go out and replace the plastic. And you have to buy additional plastic, which is more input that you had not accounted for in the end. And hopefully you hope that you have enough yield to actually make up for that loss. Our site in Jashore, Bangladesh, you can see that it's very different. I would like to point out really quickly that the yield is not as high. And once again, this is in that central part of Bangladesh where tomato production is not the main theme. The soil is very clay and it's very heavy. And here, this is the vegetable belt of Bangladesh. You can see that tomato production is much higher. But then again, we can clearly see that if you don't have any rain shelter or any grafting, you get the same results than you did when you had grafting and rain shelter. So that tells us that environment greatly impacts, how well these two treatments lead to yield loss. Also, you don't have the same amount of rain in one location and in the other on a yearly basis. And then when we had grafting only, there was a decrease than when you had both treatments. Interestingly, when we had rain shelter only in this particular site, we see that there was an increase in production. So sometimes you need to tinker and look at your site-specific location to really know and understand what's going on with your farm and to better tell the farmer on what to do with their farm. And finally, our last site, Shanhua, Taiwan, you can see that yield is much, much lower than in any of our other two sites. And this was at headquarters. Su Hongling, who was the researcher working there, he's worked on tomatoes all his life, but unfortunately that summer we got hit with 3 different hurricanes in the AVRDC. That's an excess amount of rain, and we have excess, and then we have excess of rain. And also the conditions were just really horrible. And you can see from these pictures with rain shelter, no grafting, or no rain shelter and grafting, that you barely see green in the middle of those rows. So the tomatoes just were not happy at all. And once again, grafting and rain shelter really don't have an effect on yield. There were some really interesting things that came in terms of the diseases of importance and the incidence of those. Those are very complicated, and those would need another talk in themselves. But One of the most important diseases right now are tomato yellow leaf curl virus in Bangladesh, and the AVRDC has been working tremendously and trying to breed resistant varieties for those, and they've been successful, and they're currently running trials on those in Bangladesh. Tomato fruit borer was the most severe insect, and tomato leaf miner as well. Black leaf mold is by far the most destructive disease in the field. They need to spray sometimes 2 times a week to be able to just have a crop going. And this is what your typical Bangladeshi plot would look like with rain shelter right here. It's always made out of bamboo, and you have the plastic covering on top. So conclusions, good communication is key. Grafting and rain shelter varied by location. Black leaf mold, once again, was the most severe disease in all of the plots. Tomato yellow leaf curl was a serious problem across all treatments, across all locations. However, there is resistance being bred for it. However, there's that race between pathogen and plant and host, and maybe tomato yellow leaf curl virus is being faded out, or they're actually dealing with this, but tospa viruses are coming in, and they were very, very severe during the trial period. So recommendations is to better find plot locations for some of these treatments. A lot of the efficacy of the rootstock was not really able to be addressed because bacterial wilt was not really an issue in any of the plots that we had. It was just not in the soil. It would also be conductive to conduct some of these experiments with the wild species of rootstock that the Bangladeshi people already use. And very important to resupply the food chain. What's the point of growing more tomatoes if we don't have somewhere to sell them, and that profit actually coming back to the farmers? Use other techniques, not necessarily the rubber tubing, which has to be imported into the country to actually do some of the grafting. Our colleagues in India use straws, for example. And another suggestion was to try to use plastic mulch to cover some of the plots to see if that would be sufficient. So then, some food for thought. Sometimes we all think of technology, or how can we use our resources? And it's all location specific, I feel. So here, this is right after a really short rain in Bangladesh, and you can see an excess of water and the vegetable vendors and the market back here. And if it was any of us right now, we'd probably like, oh my gosh, I can't even walk through there. But, it's a normal day in Bangladesh, so you just walk. And if you're really smart, you take your soap and you go where there's a gutter, and you just wash your hair, and you just wash your body, you dry off, and you go off, because that's water that you don't have. And with that, I would like to thank you and take any questions if there's time. Yes, Ana Maria.
Do you think that the grafting had a detrimental effect on your GSR?
Yes, I definitely think that the grafting had a detrimental effect. If you are not sowing, or if you're not planting in a field where bacterial wilt is a problem, you will have delay in growth. It takes much more energy to just grow a tomato when you use grafting. So there's definitely an impact. And if you don't need it, don't use it. Yes, in the back?
You did show that what are the other diseases that you were able to see in tomato, like fruit borer and UILC, right?
Yes.
I was just wondering, how about the fusarium wilt?
His question was, did I see any fusarium wilt in some of the field trials that I conducted in all 3 sites? I did see some fusarium wilt. It was not high. Maybe 2 or 3 plants in a plot that I saw with fusarium wilt. But in this case, I was very lucky. I think a lot of that maybe has to do with the EG203 that has some resistance to fusarium. Yes. This question is, did I see any Phytophthora late blight? I did not. I did not see any. I tried to document wilting. Again, the thing is, I wasn't there all the time, and I usually just took data from my colleagues. When I was in Bangladesh, I did get to go to the field a couple of times, and we would take samples back, but then I would have to go back, so it would remain on my colleagues to finish that work, and didn't come to my attention that if we did or we didn't. So I can't say definitely no, we didn't have any, but no, none of it was documented. Yes.
Are you working with an economist? Do you have an economist collaborator?
Do I have an economist? Yes, I have an economist collaborator at the ABRDC, and he will actually be doing the cost-benefit analysis of these. And this research is actually going to be conducted again with some of the recommendations from the guinea pig. Yes.
We can continue with the questions later. We need to move on. Thank you, Antenna. Okay, our third speaker is Anna Teston. She's coming from Ohio State University, and she will be talking to us, making sure your title is the same, about seeding a participatory soil and plant health program in Morogoro, Tanzania.
I'd like to thank the organizers of today's symposium. Good afternoon. Mimi ni mwanafunzi wa magonjwa ya nyanya za Tanzania. In Swahili, I just said I'm a student that studies tomato diseases in Tanzania. And I've been in Tanzania for the last 3 months working on some PhD research as part of a collaborative research program between Ohio State University, Sokoine University of Agriculture, IITA, and the Tanzanian Ministry of Agriculture. This research is funded as part of a project under the iAgri, or Innovative Agriculture Research Initiative, which is a program of part of USAID's Feed the Future initiative. Now, within Tanzania, we work in the Morogoro region. This is the third largest region in Tanzania, and it's also a major agricultural region. It's a large tomato producing region, and that's why we're working there for our project. Now, within Morogoro, we work in 5 villages: Misufini, Mabana, Misangozi, Mali, and Kibagala, and our main collaborators are located at Sokoine University of Agriculture in Morogoro town. Now, the farmers that we work with grow tomatoes as their main cash crop, and this is the 3rd tomato talk in a row, so I think you understand that tomatoes are a very important crop in these countries. Now, tomatoes also provide an important source of nutrients, and in these villages, our farmers are growing processing-type tomatoes, so they grow a medium-sized, plum-shaped tomato. And these tomatoes are usually rotated with either maize or rice. Now, there's constraints to tomato production, and there's many constraints. So in our research, we focus on 3 main constraints. We want to address losses from diseases, we want to improve access to improved germplasm, and also we want to address losses from poor soils and poor soil management. We also asked the farmers that we work with what they thought the key constraints to their production were. And overwhelmingly, our farmers responded that diseases and insects most greatly limited their profitability, followed by a lack of market access. Now, fewer farmers responded that unproductive soils and unproductive varieties limited their access or profitability. Now, the overarching question of this research is, can we develop methods to improve soil health and tomato disease management for smallholder tomato farmers in the Morogoro region? And to do this, we're integrating soil and plant health, and we're taking a participatory approach in which the farmers are involved at every step of the research. And so today, I'll talk first about the participatory plant health aspect of our research. I'll go over what we found in a disease survey of tomatoes in Morogoro. I'll then move on to talk about what we learned about local plant disease knowledge, and then I'll discuss some tomato variety trials that we have in place. I'll then switch gears and talk about the participatory soil health aspect of our research, and I'll discuss how we're trying to improve the ability of farmers to evaluate both local and technical indicators of soil health. I'll wrap up by discussing how we're trying to evaluate these programs that we have in place. So I'll first talk about participatory plant health and how we're trying to improve that for our smallholder farmers. And I want to start with this picture. Now, this is something we typically see in fields that we visit. Now, this picture, you can see the pesticide residues on both the leaves and the fruits. And this is because the main management strategy for our farmers is to spray pesticides. Now, These fungicides are oversprayed up to 24 times a season. They're sprayed for the wrong diseases, including bacterial and viral diseases, and they're sprayed without any protective equipment. Now, in the long term, we'd like to offer better management strategies for tomato diseases to our farmers. But before we can offer better management strategies, we need to have a better idea of what diseases are most common. So to get this, we conducted disease surveys in both the dry and wet tomato production seasons. And in these surveys, we sampled 10 fields in each village. So we had 5 villages, so in each season, we sampled 50 fields for a total of 100 fields that were surveyed. Now, within each field, we sampled 3 2-meter square plots for both disease incidence and severity. And this is what we found. For fungal and oomycete diseases, we found late blight, early blight, alternarious stem canker, septoria leaf spot, and various fungal root and stem rots. Here you can see Sclerotium rosei. For bacterial diseases, we found bacterial spot, bacterial wilt, pith necrosis, and we also found the phytoplasmal disease, big bud. For viral diseases, we found tomato yellow leaf curl virus, likely accompanied by other Gemini viruses. We also had confirmed cases of other common tomato viruses, including potato virus Y and tabamoviruses. And also we found quite a bit of root knot nematodes. Now, it's also important to know how often we found these diseases. And this is a graphic that shows the incidence by field in each village. Now, the way to read this graph is that the diameter of each circle indicates the number of fields in each village that we found the disease. And the color of the circle indicates what disease we found. Now, it's a bit too much to look at now, but let me walk through what we found. Now, the most commonly occurring disease that we found in both seasons was early blight, followed by bacterial spot, septoria leaf spot, and also viral diseases. Now, during the dry and wet season, we had a difference in what the 5th most common disease was. And during the dry season, we found gray mold. And in the wet season, we found a lot more tomato yellow leaf curl virus. And when you put up all of what we found, you can see it's a lot more complicated of a picture. Now, for truly devastating diseases of tomato, we only found late blight in one village, Mlali, during the wet season. And we only found bacterial wilt also in one village, but during both the wet and dry season. We found quite a bit of root knot nematode, but we likely underestimated how much root knot nematode is actually present, and this is because farmers don't like you to rip up plants from their fields. Now, we also wanted to learn from our farmers what diseases they thought were most common in both the dry and wet season. Now, to do this, we gave our farmers cards that had pictures of symptoms of 20 different tomato diseases, actually 19 diseases and 1 disorder, and you can see the pictures at the bottom of this slide. And what our farmers responded was that for both seasons, they had common responses for 4 diseases. So bacterial spot, root knot nematode, tomato yellow leaf curl virus, and late blight were reported to be most common and most important for both seasons. Now, we agree that bacterial spot, root knot nematode, and tomato yellow leaf curl virus were commonly found in our survey, but we only found late blight in 1 village. Now, the reason that we think this topped our farmers' list for both seasons is because when late blight does occur, it has very distinctive symptoms, and it has a truly devastating effect on yield, which makes it a very memorable disease. Now, another important piece of information that we gathered from our farmers was what they called these diseases in their own local language, which is Swahili, and there's also other local languages spoken. Now, to do this, we had our farmers broken down in groups, and they used those same 20 cards, and they would tell us the name of the disease in Swahili, and then they'd give us a few words on the symptoms of the disease. And from these disease names, you can learn quite a bit about what farmers know or observe about different properties of the diseases. So you can learn what farmers are observing about epidemiology of diseases. So late blight in Swahili is known as fuli, and this is a local name for cloudy, drizzly weather, which are the disease-conducive conditions for late blight. Most often, disease names refer to the signs and symptoms of the diseases. So powdery mildew of tomato is known as majivu, which means ashes. Now, sometimes you can get some really interesting feedback from the farmers. And we did this in 3 villages, and for cucumber mosaic virus, we got 3 very interesting responses. In one village, CMV is referred to as fat disease, and that this disease is always associated with aphids. And CMV is an aphid-transmitted virus. Another village called CMV, sweet potato disease, and this is because the leaves of infected plants are very distorted and resemble those of sweet potatoes. And this is what happens when a tomato is infected with CMV. The leaves become very distorted. A third village called CMV, female leprosy, because only small fruits are formed, and when a plant is infected with CMV, either no fruits or only small fruits are formed. Now, this is all very interesting from an ethnophytopathological standpoint, but we also want to be able to practically use this information. And so we're using these local names, and we're including them in disease identification and management cards. Now, these cards have pictures of key symptoms of the diseases and also simple management strategies. And we'll also include the local name so that when a farmer looks at the card, they can recognize their local name and better recognize the disease that they're working with. So I went over what we learned in the disease survey and local knowledge, and I want to move on to some participatory variety trials that we have in place. And the basis of any good disease management is the inclusion of varieties that have good levels of disease resistance. So in these trials, we're evaluating different tomato lines to see how they perform under the disease conditions in Morogoro. And when you use participatory variety selection trials, farmers are immediately involved in the variety selection process, and this has 2 benefits. First, it decreases the time needed for variety selection because you're not wasting time on varieties that farmers are not interested in, and also it increases variety acceptability because farmers are gaining valuable experience with these varieties from the beginning of the experiment. Now, we're using a type of participatory variety trials known as mother-baby trials. And these trials are being conducted in 3 villages, and we're evaluating 3 new test varieties, which include 2 Ohio State inbred breeding lines. These are processing-type tomatoes that have high general levels of general foliar disease resistance, and also MT56, which is a bacterial wilt-resistant line of tomatoes. And we're comparing these test varieties to 7 commonly grown local tomato varieties. Now, as I said, we're using the mother-baby trial design, and this is a unique type of trial design that was originally developed to test soil conservation management strategies. And in mother-baby trials, you have 2 types of trials. You have larger mother trials, and you have smaller baby trials. Now, mother trials are set up as commonly like on-station trials. They're randomized complete block designs, and they include all your treatments, which in our case are all the varieties. So in these trials, all the 3 test varieties that we have are compared to the 7 local varieties. Now, in these mother trials, these are held in the village that you have the baby trials, and these trials are run by farmers, but management is dictated by researchers. Now, the data that you gather from mother trials is quantitative data on crop traits such as yield and disease incidence and severity. Now, at the same time you have mother trials running, you also have baby trials. In baby trials, you have about 10 to 12 in a village. Now, in baby trials, these are small randomized design trials, and in these trials, individual farmers are comparing the test varieties to their personally preferred local variety. Now, in these trials, management is solely up to the discretion of the farmer running the trial, and from baby trials, you gather data on qualitative crop performance. So at the end of the season, you ask the farmer to rank the tomatoes on rank the different varieties on different characteristics, such as yield or disease incidence. Now, what you're trying to get from the mother trials is good quantitative data on crop performance. What you're trying to get from baby trials is data on how the farmers are accepting these new varieties. So you gather information on crop acceptability. So these varieties, or these trials, we started in June, and we have about a month and a half left in them. But I have some preliminary data to share with you. And this is data on early blight incidence, which, if you remember, was the most commonly encountered disease in the disease survey. And in this graph, blue bars are significantly different from yellow bars, and green bars don't significantly differ from either blue or yellow. So you can see that there are some significant differences in 2 of the 3 villages that we're working in. But also, we're seeing a trend that the 3 new test lines seem to be at the lower end of of early blight incidence in all 3 villages. Now, whether or not this actually holds up at the end of the season is yet to be seen, but that's something we'll evaluate within the next month. Now, we're also getting information from our farmers that are running the baby trials, and they're giving us some pretty strong opinions on the varieties that we've introduced. So for MT56, our farmers don't like the size or shape of the fruit. It's too big and too round to pack nicely in boxes like they normally do. They do like that it fruits early, but they don't like the cluster size, which is only about 3 to 4 fruits. For the 2 Ohio State lines, they are liking these so far. The size and shape of the fruit is correct. It's a medium-sized plum. It fruits early, and it has a cluster size of about 7 to 8 fruits, which all ripen at about the same time. So I want to switch gears now and move from plant health to soil health. And as we know, if you attended the plenary session, these are very closely linked. And I want to start with this picture. And on the right-hand side of this picture, you can see sand overlaying a loamy clay soil. And the reason that this happens is because several of our villages are located on rivers that flood every year. And so from year to year, our farmers don't know what their soils are going to be like after these floods recede. So in this case, our farmer now has a field that has 2 different soil types in it. Even if our farmers are not using farms that are located on rivers, the soils in the villages are highly variable. So this is a picture that illustrates some textural differences. So in a single village, soils can range from a pure sand all the way to a pure clay. And these soils also differ in other characteristics such as pH, electrical conductivity, and nutrient levels. So the question is, how can we monitor soil health when soil conditions are so highly variable? And one thing you have to do first is define soil health. And you can use a technical definition, and we prefer that of Doran. But for the purpose of this talk, I want to just have a working definition, is that we want to have soils that can support the production of good yields of tomatoes for a long period of time. And when you're evaluating soil health, and that's what we're trying to do, is improve the ability of farmers to evaluate soil health, you can evaluate both local and technical indicators of soil health. And these indicators can be broken down into different soil characteristics, including chemical characteristics, such as pH, physical characteristics, such as texture, and biological characteristics, such as microbial activity. Now, the difference between local and technical indicators is that local indicators are normally defined by farmers, while technical indicators are usually defined by researchers. Local indicators are qualitative in nature, while technical indicators are quantitative in nature. And local indicators are usually evaluated directly in the field, while technical indicators usually need to be evaluated in a lab. Now, for this part of our research, we want to address the question of whether or not we can evaluate these indicators quickly and inexpensively in the field. And we did just that during the dry season survey. In 2013, we used a low-cost soil test kit, which was originally developed by USDA NRCS, to evaluate the soils in the 50 fields that we looked for diseases. So we wanted to also follow up with our farmers to see what they knew about soil testing and evaluation. And 73% of our farmers were aware of soil testing services, and 43% had had their soils tested before, which is fantastic. That's about half of our farmers. But it's not so good when you consider that 40% of the farmers had only had their soils tested for the first time when we tested the soils in their fields during the dry season disease survey. And the one farmer that had had their soils tested independently never received the results of the testing. Now, we also asked our farmers whether they agreed or disagreed with different statements on soil testing. And all of our respondents responded that they strongly agreed that they would like access to soil testing, but over 75% of our respondents either strongly disagreed or disagreed that they did actually have access to soil testing. So this is something that needs to be improved for smallholder farmers worldwide, is access to soil testing. So I'm going to first address how we're trying to improve the ability of farmers to evaluate local indicators. And the first thing you have to do is to identify what farmers are using to describe their soils as either good or poor. So to do this, we had an activity and we asked farmers to identify 10 characteristics of soils that are good for tomato production and 10 characteristics of soils that are poor for tomato production. And what we did after we had the characteristics from our different groups is that we would take those soil characteristics and then link those characteristics back to what indicator they were referring to. So if a farmer said a clay soil was poor, that would go into the category of texture. And what we found was that for both good and poor soils, our farmers were using 3 main indicators to describe them. Either texture or tilth, soil-water relationships, and soil fertility were used mainly to describe soils as both either good or poor. Now, there are other indicators that were used to describe soils, such as the presence or absence of pathogens, where the soil was located, and soil color. But again, it's just those 3 main soil indicators that were used to describe soils. Now again, how do you use this local information to benefit farmers? And the way that we're using it is to develop a soil health card. And a soil health card is a qualitative tool that a farmer can use directly in their field to quickly evaluate their soils. And this was also a tool developed by NRCS. Now, in a soil health card, you have, as you can see on the left-hand side, a list of indicators. And for this soil health card for Tanzanian farmers, we gathered our indicators based on responses from the activity I had just previously described. And for each indicator, you have 3 levels that qualitatively describe the soils. So you have poor, medium, and good levels. So a farmer can go in the field, pick an indicator, and then quickly evaluate it. Now, these cards are being translated into Kiswahili, and when I return to Tanzania at the end of this week, this is on the agenda of things to start evaluating in the field. Now, moving on to technical indicators. These are a little bit more difficult to evaluate in the field because usually they're evaluated in the lab, but sometimes you can bring the lab to the field, and we're trying to do just that. So we're adapting a soil test kit that was developed by NRCS, and we're using that to evaluate soils rapidly in the field, and we're going to train farmers on the use of this kit. We've selected a few tests to focus on first. So this kit will be used to evaluate soil bulk density, infiltration rates, pH and electrical conductivity, soil temperature, soil nutrient levels, and also aggregate stability. And the question that we're trying to answer here is how useful is the soil test kit as both as both an educational tool to teach farmers about different soil characteristics and as a means to triage soils. So can a farmer quickly identify a problem with their soils and then change their management strategy or change their cropping system to address these constraints? So I've gone over how we are trying to improve both soil and plant health, and now I'm going to go over how we are evaluating the programs that we have in place. And we're using a method known as outcome mapping. And this is a method to track program progress that was originally developed by the Canadian government to evaluate international development programs. And in outcome mapping, you track changes in participants' actions and behaviors during the course of the program. Now, typically, when you're evaluating international programs, you track long-term changes, but tracking these long-term changes can require a lot of resources. It's very expensive, and sometimes you can't attribute these long-term changes directly back to your program. So we're using outcome mapping to evaluate both our mother-baby trials and the soil test kit. There's 3 core components of outcome mapping that I'm going to go over some examples of the mother-baby trials to teach you more about them. So the first step of outcome mapping is to identify boundary partners. And boundary partners are the people that you want to influence with your program. So for the mother-baby trials, our boundary partners are farmers, village leaders, and extension agents. Now for each boundary partner, you identify an outcome challenge. And this is the change in behavior or actions that you'd like to see if the program is actually successful. Now, the example for farmers in the mother-baby trials is that we want farmers to participate in mother-baby trials, provide feedback on tomato varieties, and help to select improved tomato germplasm. Now, you have to track whether or not you're actually meeting this outcome challenge, And you track progress towards this outcome challenge with the use of progress markers. And with progress markers, you have 3 different levels. Those that you expect to see, these are very realistic changes that you would see even if the program is barely working. Those that you would like to see, these are very realistic changes that you would see if the program is working as it should be. And those that you would love to see, which are things that you would only see if the program is working beyond your wildest expectations. Now, with these progress markers, you start evaluating them right from the beginning of the project. And using our example from the mother-baby trials, in 2 of our villages, Misufini and Misangozi, we've seen changes that indicate that 9 of 15 progress markers have been met. While in Mabana, we've seen that only 4 of 15 progress markers have been met. And what this tells us, what this allows us to do, is redirect valuable time and resources to address aspects of the project that aren't working as well as they should be. So in this case, we know that we need to refocus efforts on Mabana to bring them up to the speed of Misufini and Misangozi. So I've gone over how we're evaluating our program, but I want to recap now the outputs of this project. Now we're creating outreach materials for farmers and extension agents, both the disease identification and management cards and the soil health card. We're introducing new tomato germplasm, and it seems like 2 of the varieties that we're introducing seem to be well received by the farmers so far. And ultimately, we would like to introduce the soil health test kit. Now we're still at the very early stages of evaluating this kit, but in the long term, this would provide a tool for farmers to rapidly evaluate their soils. So in conclusion, this research is leading to improved knowledge of tomato production in the Morogoro region of Tanzania. And because this is an important cash crop, it's important to support research that supports its production. This research is also establishing important linkages between researchers and farmers, and these linkages are extremely strong because we're taking a participatory approach in which the success or failure of this program requires the direct participation of our farmers. Ultimately, this research has the potential to improve soil and plant health monitoring and management in Morogoro. Now, in conclusion, I'd like to thank those who made this research possible. So this project was funded by 2 programs under the USAID Feed the Future initiative. First, through a collaborative research grant from IAGRI, and second, from a U.S. Borlaug Fellows in Global Food Security graduate student research grant. I'd like to thank our collaborators for their support and suggestions in this research, but there's 2 people in particular I'd like to thank. I'd like to thank my advisor, Dr. Sally Miller, for this opportunity to travel to Tanzania and for her help and support along the way. And also I'd like to thank Dr. Delfina Mamiro from Sokoine University of Agriculture for her help in all aspects in research and life in Tanzania. But I'd really like to take this opportunity now to thank the farmers that we work with. They welcome us to their villages and their field, and without them, this research would not be possible. And with that, I think I have time for questions. So thank you for your attention. So the question is, did you decide on the outcomes and the markers with the farmers as well, or just researchers? And that is just done with the researchers. You have a small team of researchers that get together and evaluate that. I think if the farmers knew, they might change their actions and behaviors to make it look better. So it's better to have somebody outside setting those outcomes and the progress markers. Yep.
So also about the outcome. So this change in behavior, is there a way that you could track that this change in behavior would remain in the population after the project is over?
So the question is, with outcome mapping, is there a way to track changes in behavior after the program has ended? That would require additional resources, and you could do that if you had additional resources. For the timescale of this project, we have 2 years, so we can't go beyond that.
Do you expect these changes in behavior to be kept in the population?
If they do accept these new varieties, I would expect that they would keep using them because it's very difficult to get new tomato germplasm in this region of Tanzania.
Thank you.
Cara?
Borlaug, how does they be providing that germplasm to farm grower or breeders
So the question is, will Ohio State provide that germplasm? So the germplasm we did provide is inbred lines, so the farmers can save seeds without seeing it. Well, they shouldn't see much of a change between seasons. And that's why we selected inbreds and not hybrids, because hybrids you need to go back to crossing every year to get that same seed. So we have already provided that support, and we are increasing seeds to give to farmers at the end of these mother-baby trials. So the question is, with the soil test kit, will we provide some further information on how to change management? Yes. So on the soil health card, on the back of that card for each indicator, we'll suggest different management strategies to improve that indicator. And also with the soil health kit, there'll be management strategies that are included, you know, if they find too high or too low of a pH value. So that's all stuff that we're working on. So the question is, do you have to provide incentives to the farmers that are participating? So the farmers have two incentives. They get to keep the seeds at the end of the season, and we provide a small per diem from taking a break during their day, because we do take up quite a bit of their time. It's not extravagant, and it's appropriate at the local levels. Thank you.
Okay, we're taking a 15-minute break, and we will resume at 2:45. All right, everybody, let's resume the session. So our third student speaker this afternoon is Abraham Fulmer, who's coming from the University of Georgia, and he will be talking to us about his research in Haiti. The title of his talk is Mountains Beyond Mountains, Challenges and Opportunities for Managing Peanut Diseases in Haiti.
Thank you so much. It's a pleasure to be here today. I'd like to acknowledge all of the folks that have been a part of this research. Dr. Kimmerate, who is my major professor, as well as Dr. Brenneman, who's on my graduate committee, and a whole host of other folks, including Imana Power, who is currently watching this from Suriname, and just want to welcome everybody to this presentation today. Mouse. Left click. In Haiti, there is a proverb that says, beyond the mountains, there are more mountains. And of course, this oftentimes expresses the idea that once you reach the pinnacle of one problem, you arrive only to reach the pinnacle of another problem. And of course, in Haiti, this is very pertinent, as in many developing countries where there has just been a long history of political turmoil and instability, economic heartache and devastation, and of course, along with that comes poverty, and environmental degradation is a major issue as well. And all of this together coming back to point to the struggle for existence in a place like Haiti, and particularly where we see the effects of poverty in terms of malnutrition, where this is particularly acute in many children. Thank you. And so this is a major problem that is in Haiti as in many other parts of the world. Now, one good thing I'd like to bring to your attention is the use of peanut butter medicine, which is known in Haitian Creole as medicamamba. This is RUTF, ready-to-use therapeutic food, that principally consists of peanuts. Well, a good portion of it consists of peanuts. And it has shown dramatic effects on reversing the detrimental issues relating to malnourished individuals, such as this young lady here. You can see what sort of amazing effect this medicamamba had for her. Now, this product is currently being produced and distributed in Haiti by a NGO called Meds and Foods for Kids, or MFK for short. And this project has been going on in Haiti for about 2006, 2007. So what has happened is that this has created a large demand for locally grown peanuts in Haiti, particularly distributed by Meds and Foods for Kids. And of course, this is now being marketed as a product called Plumpy Nut and being distributed in Haiti through Nutriset, which is buying it from MFK. So MFK has this goal of buying all of their peanuts from locally grown peanut growers in Haiti. Now, the question becomes, is Haiti capable of providing those peanuts? And I suppose it would be interesting and appropriate here to talk a little bit about peanut production in Haiti. Peanut production in Haiti is an ancient practice. It goes back to the days of Christopher Columbus and his faithful sidekick Bartolomé de las Casas. And so who knows how long peanuts had been cultivated on Hispaniola before the Spaniards arrived. But one thing that's certain is that the peanut producing methods and technologies really haven't advanced that much more in many a year. Now, it's a small crop. It doesn't come up with, in terms of land use, it represents about 1%, but it's a very important cash crop. And of course, folks there love peanuts. They're my kind of people. They'll eat them boiled, roasted in peanut butter, spicy peanut butter. Hadn't tried that yet. But peanuts are very important, and folks love eating them. But in terms of supplying MFK's demand, there's some serious problems. One is small volume. The second one would be low quality, and the third would be high cost. So I want to briefly mention some of the causes of the small volume. One is late leaf spot, and the other foliar disease would be peanut rust. Now, both of these are most likely the major contributors to reducing yields in Haiti in terms of peanuts. And most growers average around 400, 500 pounds per acre. Now, what does that mean? In 1950 in the United States, we were producing on average in Georgia about 1,500 pounds per acre. Now we're averaging about 4,000 or a little over 4,000 pounds per acre. So there's an obvious yield gap between actual yield and attainable yield. So there are some other limitations as well. Stem rot caused by Sclerotium rollsii. Mind pouring me a little water, please? And a little water, please. As well as Diplodia collar rot. We're not really 100% sure what Diplodia, how widespread it is at this moment. It was just recently reported. Thank you. But this is something that needs to be investigated in the near future. Now, many of us are aware of what causes low quality in peanuts, primarily aflatoxin contamination. And Dr. Dan Brown from Cornell University has done quite a bit of research in Haiti and has initially identified samples in averaging right at about 800 parts per billion. Now, Haiti and Meds and Foods for Kids in particular needs to be able to have a standard of about 20 parts per billion to be able to produce the medicamamba. So there is a lot of room here for working with both quality and quantity. Now, when we think about all of that together and we think about Haiti, it makes kind of sense that in the Feed the Future initiatives that we've heard about today, Haiti was identified as one of those countries targeted to reduce hunger, poverty, and undernutrition. Now, under the umbrella of this, a new grant, part of Feed the Future with USAID, is the Peanut and Mycotoxin Innovation Lab. And this is based and managed out of the University of Georgia with a mission to apply leading innovative U.S. science to improve peanut production and use, raise nutrition and awareness, and increase food safety in developing countries, and Haiti being one of those countries. Now, there's another level of complexity here. The foundation for the work being done in Haiti was put down in the Peanut Crisp Project, which lasted from 2007 to 2012. It was also a USAID funded program, and it was a multidisciplinary effort and included specialists from the University of Georgia, Florida, Oklahoma, NC State, and primarily all of those were collaborating with Meds and Foods for Kids that was located there in northern Haiti, and they had the technical individuals there that were capable of performing the technical work, as well as the factory and many of the other parts of the value chain. So one of the big efforts that was made in the Peanut Crisp project was to bring in new germplasm to evaluate from ICRISAT, which is part of the CGI-AR vision. So I briefly want to talk about first is some of the results of what these variety trials bringing in this new germplasm looked like in Haiti. Now, these variety trials were performed in Cap Haitian, which is in the northern part of Haiti, which incidentally is right about where Christopher Columbus landed many a year ago. And if you just zoom in on that area, we had 3 locations, very close proximity to Cap Haitian, where these variety trials were performed. Now, what happened is that ICRISAT sends little small packages of seed, that's not available in bulk volume, and they take, in this case, it's about 15 genotypes, and those are to be compared with susceptible standards. In this case, it was either the Tamnut or Tifgard, which are improved varieties from the United States, but very susceptible varieties that require heavy fungicide inputs, or the local runner and Valencia types. And to the best of our ability, these were done to mimic grower conditions in terms of no irrigation and no fungicide applications. So I briefly want to cover some of the results of what this looks like in terms of introducing this resistant germplasm. Now, the color coding typically indicates significant differences, but one of the things that is very obvious to tell is that all of the ICRISAT lines had much lower levels of both leaf spot and peanut rust compared to Tamnut, which in this case is the introduced variety from the United States. And now there are some numerical differences between the ICRISAT lines, but on the whole, they all show remarkable levels of reduction in foliar diseases. There are a few that typically stand out throughout for both reduction in disease and in terms of increased yield, which here we see both 36 and 46 standing out above the crowd numerically. But again, all of the ICRISAT lines performing much better, providing much higher levels of yield compared to Tamnut. Now, at the next trial at UCNH, we did not have any leaf spot severity ratings, but again, looking at rust, you can see that all of the ICRISAT lines had much lower levels compared to Tifgard in this case. So again, 36 seemed to show some little bit better across the ICRISAT lines, but all the ICRISAT lines definitely looking better than Tifgard. And again, for yield, a couple of these ICRISAT lines seem to stand out, but all of them were at least just as good as Tifgard, which again is from the United States, but most of them were numerically better. And the last trial at the Isle Adam, you can see that the leaf spot severity again was much lower for these ICRSAT lines compared in this case to the local runner and the local Valencia. And so again, just very dramatic reductions in the level of foliar diseases. Now, looking at all three of these trials together, we thought, 36 and 46, 50, some of these, 30, that looks good too. At some point you've got to decide, okay, which one of these would we want to begin trying to increase in terms for developing a much higher level of seed. This is where I want to first begin talking about some of the challenges to doing development work in a place like Haiti, because when you're in a situation where you're given a little bit of seed and you've got to start from the ground up in terms of getting that seed propagated, it's very easy to overlook the fact that in many of these projects there's just a lack of continuity. You're continually cycling in and out qualified personnel. It's a very slow process. Of course, you need multiple generations to build up enough seed, and then you just lack a secure farm sometimes. We don't have a research farm there that is always available, that has irrigation. It's just kind of sometimes the luck of the draw where you end up. And so in this case, we actually lost the seed, not actually lost it, but it was lost due to a drought, disease, rats, whatever the case may be. And by hook or by crook, we ended up with 99-030 that was left. Now, all the ICRSAT lines looked really good. It just so happened that 030 looked pretty well as good as well. So right now, this seed is actually being propagated. This whole field behind this this farmer here is all of what he's holding, which is the 99-030. Now, to put that in a little bit of perspective, on this side here to the right, this is the 99-030, and this is Spanco, which is the introduced variety from the United States. And you can just see there's a dramatic difference in terms of pods as well as foliar levels of control. So the plan at the moment is to continue propagating this seed and to get it out there as best as we can. There's about 2,000 to 3,000 pounds of peanuts behind this gentleman here, and is to get that seed out, and in the meantime to continue evaluating varieties from ICRSAT. Now, I want to shift gears a little bit and talk about the usage of fungicide applications in Haiti. Now, this is one of several fungicide trials that has been performed over the last few years, and if you look, the bars, the columns to the left indicate where 2 fungicide applications were used on one of 4 varieties. You had the local Valencia, and then you had the 3 Spanish varieties from the United States. And you can see that with all of these varieties, it's obvious that the fungicide had a very overwhelming effect. Now, this was just 2 replications, but, this was consistent across multiple locations and years. So it's not that big of a surprise that fungicides would provide that amazing of a transformation. But, you can go in 2 weeks, January 29th, you can tell these are starting to go down a little bit, but bam, by the time you get a few weeks later, you're really losing a lot of your available, your ability to carry those peanuts to full maturity. And that's an interesting tidbit, is that most of the farmers there think that it's time to harvest when your plants die. Now, what's happening is that most of the time there's still about 20, 30 days of available maturity that needs to be required there in order to get those peanuts to their fully matured state. Now, we're very much aware of the benefits that fungicides can bring. But we're also very much aware of a lot of the negative impacts or risk that come along with using fungicides. And so, we're very much weighing these options out. Fungicides are used, and pesticides in general are used in Haiti currently. But it's not a very well developed system. And for example, this individual, this young gentleman here, is, walking around in his flip-flops, similar to some of the other pictures we've seen today. He's having a good time going somewhere, fixing to spray something, or already got done. So, it's trying to figure out where do you really emphasize your impact? Where do you draw the line in terms of what inputs are really worth investigating and pushing? So in a situation here, we're very much still weighing out the options and testing that. It's not a done deal. And I just want to make a statement that PMIL, in fact, is this grant, this newer grant, is very restrictive and conservative as to how pesticides are applied and used. And so also the applicators that have been making the fungicide applications so far under this new grant receive training on both health, safety, and environmental issues, and they also receive personal protective equipment. So the jury is still out in terms of where we find a place for this in terms of our efforts there. Now, another big emphasis of the work that has gone on in Haiti under the Peanut Crisp, and what will continue to be a major impact in Haiti, or focus in Haiti, is education and grower awareness. And the fact that most growers are just not aware of the fact that they can actually do things that double or triple or quadruple their yields. It's a mind-blowing thought a lot of times. And so just basic agronomic principles, integrated disease management, and of course aflatoxin management are all major focuses that a lot of students and agronomists, as well as growers, both men and women, are invited to these seminars, 2 to 3 day seminars sometimes, and where there's a field outing component where they can go and put their hands on peanuts and their eyes on peanuts that have received inputs, or those that are grown just the way they're typically grown. And a part of this also that has been very useful is that a production guide that was actually developed in a very similar socioeconomic setting, Guyana, was able to be translated into Haitian Creole, and this has proved very useful for training efforts there in Haiti. Now, we always love being able to document the fact that we've done something useful and that it's stuck. It's very challenging to do this in a place like Haiti and some of these other places we've heard about. But I really think that we're kind of on the verge, after 5-6 years, of really just being able to reap some of those initial first fruits. And I do want to say, though, that MFK estimates that over 600 farmers and agronomists and small peanut processors have been trained since 2006. And so I thought it would be interesting to include some words from the wise for those who may be interested in proceeding in developing work, or just want to compare their experiences with some folks that have been involved from this project from day one. And that's one of them is with Dr. Brenneman. And I quote here that he says that it's critical to figure out what your role really is and how to get the most of what you really bring to the table. And Dr. Brenneman, for those who know him, is very, very much the thinker in that direction, and he's brought a lot of wise insight to that. And really I thought this was very informative when he says that the choice of how you spend your time and who you collaborate with often determines whether you will really be successful or not. And most folks here have talked about the great partnerships that they've formed, and I believe Dr. Brenneman would give a hearty amen to all of that. Now, from Dr. Kimmerite, who's my major advisor again, he would definitely be of the same opinion with Dr. Brenneman, but he also states that the language and cultural barriers, and particularly gaining the trust that has to be gleaned from the growers, is really one of the great challenges in terms of working in developing work, in terms of carrying plant pathology and knowledge to these growers. But he believes that some real impacts have been made in terms of education and grower awareness, but perhaps the single most important impact has been the introduction of these improved varieties. So as this project moves forward, the new P-Mill project will team up with multiple NGOs, and for example, Accesso International, TechnoServe, and of course Accesso is backed by the Clinton Foundation, and this is going to bring in a whole new impact on the peanut value chain, because they're bringing their expertise in terms of understanding the marketing and business side of things to the peanut value chain, and we're, on the same token, being able to bring our knowledge of how to grow peanuts and how to manage diseases to the table, and hopefully they'll be able to carry that with the resources that they have and add that knowledge to continue to advance the evolving peanut value chain in Haiti. Now, I just briefly wanted to give a few of my own reflections of being able to have the opportunity to work in Haiti. You know, I've rarely found few experiences where I can take something that I've been trained in, such as plant pathology, and blend that with something that I have a great desire for, which is namely to help the poor and the fatherless. But I have to say that, you know, there are real frustrations in working in this kind of situation where you have the passion, and as we like to say around my neck of the woods, the want to get out there and make a difference and implement change, and coming to realize that, you know, no matter how hard you try or dedicate 1 week or 2 weeks of every semester or something to going and giving your heart into a presentation at a local grower meeting, that may not have that big of an impact short term, and maybe not even long term if it was just that one experience. But coming to realize, you know, in a situation where there is truly a very deeply ingrained cultural mindset that resists new ideas a lot of times, and that it's very distrustful of what the information that you're trying to share with them, you know, that can be very frustrating. And so, as the old King James Bible likes to say, patience must have her perfect work. And I think that's very true in Haiti, is just banging at the door and gently finding ways to adjust to the situation. And I think we've seen a lot of great examples of that in the previous talks today, is, you know, learning to adapt to your environment and being in touch with reality, that every idea, every practice that we have may not be the best option immediately or right away or in the near or long-term future for a place like Haiti. So I wanted to share that, you know, the proverb that I mentioned earlier, that beyond the mountains there are more mountains. There's actually a different interpretation of that that's kind of shared by a few of the folks in Haiti I know, and that is they don't necessarily see that as an obstacle, but rather as opportunities, and that beyond those mountains there really can be change for good, and so I think I like that rendition, and I think it may actually be a little of both. And that's where I think we as plant pathologists are able to take what we have, the expertise and the knowledge that we have, and carry it beyond those mountains and to those mountains, and use that as a means of offering hope for those who are inclined or who have a desire to see the opportunities there. So with that, I would like to thank all of the folks that have been a part of this. There's really too many to name, but they're very much appreciated, and with that, if I have any time, I'll take some questions.
Did you see any toxo viruses and run into that problem?
So the question was, did we see any TOSPO viruses? Thus far, there have been no positive identification of any kind of viruses there that we're aware of. However, on the last trip, it is possible that tomato spotted wilt might be present there, but it's kind of along with the diplodia. Some of that still needs to be flushed out.
With this new project, and I'm assuming with these farmers, like you're teaching them personal protective equipment for spraying, are you going to have like a control and a population of farmers, and then see the health benefits in showing them later how much this really contributes to their health?
The question was, are we going to be able to set up, kind of a control with growers, those that use fungicides and those that don't, in terms of how fungicides or the personal protective equipment might contribute to their health? As far as I know right now, no. I think that's an excellent idea, though. Yes, sir.
How do you solve the aflatoxin problem that they have?
That is a very much debated question right now. The question is, how do you solve the aflatoxin problem in Haiti right now? What we have done, and I think the thing that's been very effective, is teaching sorting, proper storage, and ventilated, covered buildings, using mesh bags. But there is an issue right now with, the more you sort, the more of a concentrated, highly toxic level batch of peanuts that you have. And Dr. Dan Brown, who is main one at Cornell University looking at a lot of this, has been looking at alternative methods for how to get rid of that extremely concentrated batch of peanuts and or contaminated peanuts. And thus far he's looked at making charcoal bricklets out of it, which, if you know much about Haiti, the charcoal is the way to cook, and charcoal could be a potential, but in terms of whether peanut waste could provide enough biomaterial for that is kind of to be in question. But one of the more promising ways, too, is looking at it for oil. Haiti really doesn't have a whole lot of high-quality cooking oil, and so it's very possible that the contaminated peanuts could be used for that. The question is whether it's wise or whether we have given much thought to using multiple, not trying to get away from just introduction of one single genotype, but whether we should use multiple genotypes in terms of managing resistance. It's a great question. I may not have time to go into it fully at this moment, but the question, the answer more or less is yes, we have given thought to that, but the type of resistance, the best we know about it, it's a multigenic recessive resistance that seemed to show extreme levels of durability in the places where it has been introduced. And also, we're talking about such a small scale at the moment that we're not envisioning this being mass produced or disseminated throughout the entirety of Haiti at the moment. And it may, they've kind of got their own seed handling system, and once it gets into that, it's definitely going to be, a lot of it's going to be mingled, seed purity. That's a major issue, and it's not realistic to think that they'll be growing one single variety because they really have no way of keeping all of that seed pure. But a great question, and I may have some more thoughts on that if you hang around. So. Thank you.
All right, our last speaker for today is Tom Cresswell. He comes from Purdue University, and he will be giving a talk on reading agrios in Kabul, agricultural science capacity building in Afghanistan.
Thank you for the invitation to be here today. I wanted to start with, all right, I'm having the same problem. Are you using the mouse or the clicker? All right. This is Darul Aman Palace. It stands about 16 kilometers outside the old city center of Kabul. It's a European-style palace that was built in the 1920s by the king at the time in his effort to begin to modernize Afghanistan. Kabul was once called the Paris of Asia. There was an electric trolley car line. Education was increasing. There were considerable freedoms. It was just a plum assignment for foreign service people who were going there. But for me, this is what it looks like today. This is how I saw it when I went there. And for me, this has become a visual metaphor for what was lost in Afghanistan over the last 30 years of war. Not only loss in terms of infrastructure and buildings and the things that were bombed out, but also loss in intellectual capacity. Those people who had marketable skills, those who were able to go somewhere else during this period did leave. So there is a very large gap of knowledge in the university systems in Afghanistan. When work was begun there again in 2002, virtually all of the professors in the Afghanistan university system were at or past retirement age. So there's almost all the younger people had left, or they were just missing. So there's a very large knowledge gap there. And so this is what I've always thought of as a metaphor. If you think about sort of a brief review of the Afghanistan timeline from the Soviet invasion in 1979 and the Mujahideen resistance during the 80s to the withdrawal of the Soviets, finally with the Afghanistan being taken over by the Taliban in the 1990s, and then with the U.S. moving in and the NATO troops coming in 2002. This is just to remind you of the history recently that's happened there. There has been many different efforts at rebuilding Afghanistan since that time. The many millions and billions of dollars have been spent there, but the most recent example of that is the 2012 Tokyo Donors Conference, in which more than $16 billion were pledged by donors over 4 years' time, with Japan, Germany, and others leading the way. Canada, Australia, the U.S., Great Britain, Finland, India, many other countries have donated in many different areas to rebuilding Afghanistan. And there have been many different projects there led by governmental agencies and NGOs. Afghanistan, a picture of Afghanistan is that it's about the size of Texas. It's a very primarily an arid region. There is a lot of variation in the climate and the agriculture there, but more than 75% of people live in rural areas, and they depend heavily on agriculture. And agricultural is responsible for more than 50% of the gross domestic product every year. So it's normal that rebuilding efforts, many of them would largely focus on agricultural redevelopment and trying to improve things in the agricultural sector. There have been many efforts in many areas in Afghanistan, from NGOs to land-grant universities participating. Purdue has been a leader in that area in many areas. There have been several different projects. I'll talk about those. And I wanted to give an example of one nonprofit agency. So I'll just cover those very briefly here. But it's impossible to get a grip really on all of the different projects that have gone on in Afghanistan, but there have been many and varied. One that I'd like to highlight here has been CABI's PlantWise program. CABI is operating PlantWise in many different developing nations around the world. Many of you are probably already familiar with it. But I contacted them about their activities in Afghanistan before coming here to give this talk to get an update on what they were doing currently. The PlantWise program is focused on training local people to be, quote, plant doctors, to give them a week of intensive training about plant disease and insect problems, and then set them up with materials in the local language to conduct plant clinics in a local bazaar market on busy market days during the growing season. And then they are able to give farmers a prescription or help identify the problems that they have. And then CABI benefits from that in that they get the information back from them. That is entered into a worldwide database of information about what problems are being found in that country. So it's a cycle of farmers bringing things to the markets, the advisors there write prescriptions and then record the information that's being found, and then new materials are developed as they go along. So far, CABI has trained, has developed about 15 or so of these advisory sheets, these sort of single-page extension-type handouts, and several different publications on disease and insect management for the area. I think they've trained in the neighborhood of about 80 different plant doctors for the country. So that seems to be very encouraging. So that's one example of an NGO or nonprofit operating in the country. And then I wanted to spend the rest of the time talking about some of the programs that Purdue has been focused on in support of capacity development there in the agriculture area. Most of the programs have been USAID or USDA or USAID sponsored, and there have been collaboration with several other land-grant universities, University of California, New Mexico State, University of Maryland, and several different others. I don't have the entire list with me at hand, but it has been a collaborative effort in many different areas. The focus has been on building capacity in universities, both with their faculty there and in working with governmental agencies. The Ministry of Agriculture, Irrigation, and Livestock is the main governmental agency, but also the Ministry of Education, and there have been training programs that have taken place in country in Afghanistan, as well as bringing young professionals from Afghanistan to the U.S. for training in various areas. This is a list of all of the projects that Purdue has been involved with, and I can't cover all of them, or I don't even really know the details about all of them because there are so many. But the ones here in blue have been university focused, and the one that I was first involved with is the third one there, the Advancing Afghanistan Agriculture Alliance program that ran from 2007 to 2009. The Afghan Merit Scholars Program has already wrapped up, but we are continuing to bring Afghanistan young scholars to Purdue. There have been more than 200 individuals brought to Purdue's campus for either short-term training or long-term training. About 30 of those have been at Purdue for degree programs, either master's programs or PhD programs, most of those in agricultural fields. This group of individuals was our first work, and I wanted to emphasize that this has been a joint program with the It's really been a joint effort at the Purdue Plant and Pest Diagnostic Lab, because while I have been doing training in Afghanistan, this program happened at the Purdue Diagnostic Lab in part, where this group of individuals got training in plant disease diagnostics for a 7-week intensive program. They also received training in other parts of the day with insect or entomology and also weed science. They also had a little bit of training in ESL because their English was a little bit rough. But we really enjoyed working with this group of individuals. They're very promising, very bright. They also had some knowledge of plant pathology when they come, or some knowledge of entomology when they came. But we learned from them, and they learned from us. So it was a joint effort. And while I have been going to do some training in Afghanistan, the clinic had to operate in my absence, and that's where Gail Rule comes in maintaining that work while I'm gone. The woman in this group has gone on to work on a master's degree in entomology at Purdue, and the person to her, standing to her left, is working on a PhD in India right now, and the other three have gone back to Afghanistan to work in their respective institutions. In 2010, I went to Kabul to deliver an introductory course in plant pathology. So in 2 weeks, I delivered our entire 301 plant pathology course to a group of 27 faculty members at Kabul University. They came from 8 universities around the country, and we did both in-classroom lectures and laboratory work as much as we could cobble together given the existing facilities. We had 2 good microscopes and several moderate microscopes. We did get the autoclaves to work, and we were able to do some work with media and isolations and things like that. But the point is that we were able to give this training to them. I left them at the end of the training with flash drives containing all of my PowerPoints, all of my lecture notes, more than 200 extension-like PDF publications, and with the ability to take this and incorporate it into their own teaching. These are all faculty members at universities, so they had a ready-made package to take this and incorporate it into some of the training in their own courses if they wanted to do that. Here they were in the lab working with nematode extraction and beginning to learn about fungal morphology. In the private sector, Purdue has been involved with the Purdue Improved Crop Protection System called PICS. This is a system, well, if you take a look at this, this is what crop storage might look like in a typical Afghanistan village. A large woven basket with some kind of adobe-like material around the outside would protect the grain. And as you can imagine, the insect losses here would be enormous as weevils and other things get into the grain. Well, the thing that we've developed at Purdue over the years has been a 3-layer bag. The first 2 layers inside are gas-impermeable polyethylene, and the 3rd layer outside is a woven, tough layer that allows the bag to be handled without tearing the inner layers. And they found that at first this research was for cowpeas in Africa, and it's been widely distributed there and been shown to be very successful. And they found that it works in Afghanistan for wheat storage as well. If the grain is stored inside and those bags are sealed very tightly, then that hermetically sealed grain, it reduces the loss to insects very much. And so it's reduced the amount of losses. But more than that, these bags are being locally produced by a company in Afghanistan now. So it's also benefiting the local economy there. The work with the Ministry of Agriculture, 3 different projects there. One is an extension project helping to train extension workers. A second one is trying to develop a data collection and utilization system similar to our ag statistics project that we have in the U.S. But the one that I've been involved with most has been the Afghanistan Sanitary and Phytosanitary Project. And with that, we had a component of that was involved with training on food safety and food microbiology that was conducted by another researcher from Purdue, and also someone from our vet school conducted training on animal health and also PCR, because there was quite a bit more advanced knowledge in the animal health areas there, and they wanted some training in PCR, and he went over to do that. My part in that was working in plant health training and diagnostics training in 2012 and 2013. And then we went on to do a train the trainer component and turning the training over to them ultimately. We went to Kabul in 2012 and conducted trainings on a broad range of topics related to plant disease diagnostics. And here we tried to shift away from so much of the lecture in class to doing as much as we could hands-on. We worked with fungal pathogens in the laboratory. We were very lucky to be able to work in an animal science facility there in Kabul that was fairly well equipped. They had working autoclaves, laminar flow hoods, and several pretty good microscopes. We also carried over some additional microscopes, petri dishes, and media. We were able to make media. I was able to show them how to pour media, how to make isolations aseptically. We worked with bacteria while we were there. So it was a very productive 2 weeks, and because so much of it was hands-on, I think that they really appreciated the training. This is what they had been asking for. They kept asking us for more hands-on materials. We carried many of the compendia series with us. They have Barnett and Hunter in hand now. Each one of the participants had the standard fungal ID that we all started out with, Barnett and Hunter. So we worked with that. We also had Dan Eagle, the vegetable pathologist from Purdue, who's in one of the southern stations, go with us to help with the training, and he did a lot of good work with training on vegetable diseases while we were there. And Dr. Rick Foster from our entomology department went with us to do training on entomology. And here he is explaining insect anatomy and gave lectures explaining basic insect taxonomy, how insect orders are classified, and they were able to dissect insects while they were there. And I think they really got a lot out of this training. That was in Kabul in 2012. And we actually did, sweep nets in the field. We found downy mildew on alfalfa outside while we were there. We found canker diseases on the fruit trees in the facility surrounding this. We worked with the things that we found around us there. We carried some prepared slides for demonstrating disease problems while we were there. But it was a very rewarding experience to be there. We talked to them about sample collection and the importance of record keeping, what they were finding, and how to start tracking what they actually have in their country, because right now there are very few records about what diseases are actually found in Afghanistan. Later in 2012, in October, we carried the same set of training to Herat on the western part of the country, which is near the Iranian border. This is a photograph of the group there. In this training, Dr. Rob Wick from the University of Massachusetts, who runs the diagnostic lab there, went with me to do the training. And we had the benefit in both cases, both the Kabul training and the Herat training, of an excellent translator. This fellow here who worked with us, he has a master's degree in plant pathology from Hungary, so he was a very capable translator for us. One of the major cash crops in the Herat area is saffron. They grow saffron crocus there, so we saw some of the disease problems there, and it was very interesting to be able to work with a crop that I'd never seen before. And I brought home saffron, so it was very nice. We also did some training with extension workers there while we were there on how to collect samples, how to prepare them for sending to diagnostic labs. The labs were less well equipped at this location. It was quite a challenge to have access to the microscopes that we needed, but we were able to introduce them to the concepts and get them started in the training, and working with things like immunostrips and showing them the concepts that they needed to know. Ultimately, I think that they really benefited from this. We gave them the same kinds of materials that we'd left the other group with. And this facility in Herat now has a new building, and they have autoclaves, they have new microscopes, they have laminar flow hood, and other equipment that they need to open a basic diagnostic lab. So some of that work is now being done there with the training that has been provided. I won't say that they have a fully functional diagnostic lab, but they do have the equipment and the facility there now. And some of the work, I think, the beginning is there. They have the rudiments of what's needed. Ultimately, we shifted it toward the end of the project toward a train-the-trainer focus, away from us going to do the trainings in Afghanistan toward empowering them and enabling the most promising individuals that we identified in these trainings to do their own training. We through several calls, we identified what we thought was really needed. We took all of the PowerPoints that we developed both through my work with the universities in 2010 and through the work that we had done with the training of the Ministry of Agriculture people in 2012. And we worked with the most promising translators that we had, both in entomology and plant pathology, and all those were translated into Dari. A few of them were also translated into Pashto because certain regions of the country, Pashto is the primary language, and it was needed there for certain crops. But mostly it was Dari, a version of that we needed there. Then we also took those PowerPoints and broke them down into single disease extension type handouts. One sheet, one disease with photographs and simplified descriptions of the problem. And those were also translated into Dari. So in the end, they ended up with about 30 different PowerPoints and more than 100 extension fact sheets to work with in this training. Then those promising individuals did a pilot project with this in Kabul with evaluators there from the USAID sponsorship program to see how it was going to run. Then they took those training materials and did trainings throughout the rest of this past spring in Kandahar, Herat, and in Mazar-e-Sharif in the northern part of the country. And this is the part of the program that I'm most proud of. I'm proud of the work that I've done there over the years. I'm proud of being able to go there, but what I'm really proud of is that in my absence, the work is still continuing there, and that Agrios is still being read in Kabul even when I'm not there. So with that, I'll stop, and if you have questions. Yes?
Yeah, you mentioned that, how important it was at various sites to have good microscopes. Will it, do you think, be a great help for the programs there? You may be aware that there's a technologist in India who developed a very serviceable, very good microscope, out of folded cardboard, really. Plastic lenses, they're about $8 each, and they have, working together with just a standard cell phone, you can actually have a quite serviceable portable microscope.
I've heard of some of that, but I haven't seen it yet. I'd be very interested to talk to you about that. The question was, am I aware of the very low-cost technology for working with microscopes? And I would be very interested to learn more about that. Yes, Alejandro.
With the theme of this meeting, I guess, being impact, I'm just wondering, how are you measuring impact when you go for such a short period of time and you sort of just drop the technology and you do these really intense classes? Do you have any type of, follow through via Skype or, email with some of these people?
Well, the question is, how do we measure longer term impact when this is such a short-term project? We measured impact while we were there. We were doing pre- and post-tests for all of the trainings that we did. However, that's a different matter than measuring long-term impact. And in each of the trainings, I've always offered to Skype with them, to accept images by email for further interpretation to try to help them identify problems. I haven't gotten a lot of takers on that. Unfortunately, we have had a few cases where melon problems were emailed to us, and we were trying to help with that. But I don't know what the reason for that is. So I hope that they will continue to communicate with us in the future, and that we will continue to be able to help from afar. But one impact, I think, is that there have been more than 200 people trained at Purdue, either short-term trainings or long-term trainings. And I think that will have moved the education system at the universities forward in some measurable way, that it really has been changed. And I think that is measurable change in the university system. But maybe not, maybe there hasn't been the kind of documentation that you're thinking about. But. Yes.
How long has our impact been in Afghanistan? I mean, we've been at war there forever, but how long have we been doing this kind of work?
The question is, how long have we been doing this kind of work in Afghanistan? And the first project that I have knowledge of started in 2002, as soon as the first NATO troops had moved into Afghanistan, as soon as it was safe to enter the country. There's a professor at Purdue who had spent 17 years as a Peace Corps volunteer in Afghanistan, and he was fluent in Dari and knew a lot of people there, and he was able to start interacting as soon as he was able to reconnect. So
he's had pretty good history there for the last dozen
yes,
years.
yes. And that's one reason that many of these projects have come to Purdue, is through his efforts and experience there. Yes?
So I know you mentioned saffron as being a cash crop for Afghanistan, but through your work there, I mean, what do you see for the near and the long-term future? Because the number one cash crop in Afghanistan is still poppy. And when you read about what the farmers say, they're going to grow whatever they can get the most money for. And right now, because of the only true value chain that's sustainable right now in Afghanistan is for poppy. What do you see? How can these types of projects move forward without any other value chains?
The comment was that you think that poppy is still the number one cash crop throughout Afghanistan. I would beg to differ there that that's not really the case, that wheat is the primary crop in Afghanistan, that poppy is often grown in areas where the farmers are forced to grow it, and in some because of where they're still controlled by the Taliban, and that poppy is sometimes grown in areas where it's grown as a hedge against crop failure in other crops. And that if we can improve the dynamics of growing other crops and improve the success of other crops, then there would be less incentive to grow it in other areas. And so that's my take on it. But then I'm not an expert in that situation or that area. But I don't think that it's accurate to say that it's the main cash crop throughout the country. Yes? Oh, the participants. What participants are? Well, in the what percentage of the participants of our programs were female? In the university training, we had probably about 10% were female, and in the Ministry of Agriculture trainings, probably about 5 to 7% were female. It's just the nature of how many were employed by the Ministry of Agriculture at the time. Marty.
Do you sense any concern about with the fact sheets that we're putting together with the USDA and the branding on those sheets?
Oh, was there a concern about USDA branding on the fact sheets? I did not get any feedback about that, but we did give them the, they are provided in all formats, so they could be modified later, I suppose, as necessary, if that's a concern, I think, later on. But that was part of how it needed to be put out.
I understand that.
Yes?
What is the
what is the percentage of agricultural students in the university system? I don't think I really know the answer to that. I know there are quite a few because when we were in Kabul University, the agriculture university part, the part of the university dedicated to agriculture seemed to be quite sizable. I just don't know the answer to what percentage it is, though, but it is quite important in the university system, even in Kabul, in the city. And I think it's even more important in the other universities in Herat and other areas. I could get some more information for you if you'd like.
During your work there, since 75% of the population is rural, how much of the information do you think gets distilled from the university system into rural areas?
I think that the question is how much of the information can get from the university system to the rural areas. I think that's a real challenge, and that's one of the reasons for the project to help try to establish an extension system, because really none existed before. And I think there is some success in that area. I think they're making some progress, but I think there's a very long way to go to get that out there. The Ministry of Agriculture helps in some regard there, but I think probably the sphere of influence of each university is fairly small at this point, but there still needs to be a lot of work in that area. The question is, we created a lot of materials, observations, and we created a lot of written materials, and did we find any problems with the literacy? That's a very good observation. We were targeting primarily the people we were working with in the Ministry of Agriculture. And those people are educated in their native language, and they were that's the target for these training materials. But it's a good point that they would then need to take these kinds of materials and simplify them even further and provide more pictures perhaps. But we did simplify pretty greatly the language involved on those materials. They're not very technical. I know they're printed in Dari here and you couldn't read them, but they are quite simplified materials about just basically what is this, how is it spread, some very brief statement about how it's controlled. So I think they could take those materials and modify them further for use for actual farmer use. But no, they weren't just, we didn't create any simply just all picture materials for use with farmers. But I'm sure that could be an issue. Thank you.
Okay, let's give a last round of applause to all of our speakers. And if you would like to, yeah. If there are any more questions, well, first of all, thank you to the people watching live and to the people who will watch this later. And if there are any more questions for previous speakers or also for Tom, we can stick around for some more discussion. Thanks.